Just a little introduction. The idea of this webinar today will be to talk about how ultrasonic washers work, what is the phenomenon that helps to clean our medical devices by using ultrasonic washers, which is actually the cavitation, how important it is to test the cavitation energy, standards and recommendations that support this monitoring and this corroboration of the cavitation energy, and at the end, the solutions that we offer at Terragin for monitoring this energy and how to automatize the reading of these indicators thanks to our latest launch, which is an auto reader for specifically measuring the level of energy delivered to the indicator so as to automate, digitalize, and be able to trace down these results to a specific washer. Okay, so we will see some scientific and technical concepts about cavitation energy and how the ultrasonic washers work and how we are able to monitor it very simply and very independent of human interpretation so as to be more precise in the results. Okay, so I did not introduce myself, but I think some of you already know me. My name is Antonella Strigotti. I belong to the product specialist and technical educator team, and I've been working at Terragin for more than 13 years now. So it is a very big pleasure to be able to give you this webinar and to introduce these concepts to all the attendees. So let's start, okay, with the first very well-known sentence, but never a disadvantage to reinforce the concept that cleaning is so important that if it fails, the sterilization will fail. Yeah, cleaning is the first critical step in medical device reprocessing. Any failure at this stage compromises the effectiveness of subsequent sterilization, putting the entire reprocessing cycle and ultimately patient safety at risk. So even though we will talk exclusively about ultrasonic washing, this is the same for every different type of washing processes. And which are these type of washing processes? We have three modalities for medical device cleaning. We have manual, we have an automated washer disinfector, or we can have an automatic ultrasonic cleaner, which is the aim or the focus of this webinar. So with the manual cleaning, we have the mechanism behind is the operator dependent, brush and sink based. It is best for initial gross soil removal and delicate items. The drawback of manual cleaning is that it is highly subjective, variable consistency, and lacks objective validation data. On the other hand, we have automated washer disinfector. The mechanism is high impeachment water pressure and chemical action, and should be ISO 15883 Part 1 compliant. It is best for general surgical trays and rigid instruments, and the advantage of using automated washer disinfectors is that it delivers quantifiable thermal disinfection via the A0 values and mechanical consistency. And of course, thanks to this, it is very easy to monitor the parameters or the performance of the washer disinfector. So this is very positive for a good monitoring strategy of the washing steps. And after, last but not least, we have the automated ultrasonic cleaner, where the mechanism is a high-frequency sound waves producing mechanical cavitation. It is best for complex geometries, blind holes, fine crevices, and ophthalmic tools. The advantage of ultrasonic cleaning is that it penetrates areas that standard washer disinfectors cannot reach. Well, so for those small crevices or very complex geometries, ultrasonic cleaning helps to detach any possible organic residues in this specific hard to reach part of the medical device, and it helps to remove this type of organic residues. Okay, so we have two big type of washing processes: the manual and the automated. And as I have mentioned, the manual cleaning can be systematized, but the process parameters cannot be controlled because they are very human dependent. But on the opposite side, we have the automated cleaning that the process parameters can be perfectly automated and, as a consequence, monitored. And from the two automated ones, we will focus on ultrasonic washers because we can actually measure the parameters of the performance. So beyond the scrub brush, we have the ultrasonic washers. So ultrasonic washers are used for fine cleaning to remove soil from areas difficult to clean by other methods, such as joints, crevices, lumens, blind holes, and complex geometries. Of course, we have many manufacturers nowadays that offer different type of technologies, multi-layer or multi-level devices with different type of sizes, of course, volumes, and with or without irrigation. Okay, so we have nowadays a lot of different configurations of ultrasonic washers, so it makes even more important to be sure or to be certain that the cavitation energy delivered within the full tank of the ultrasonic washer is actually working at the right level to clean our medical devices. So, which are the components? Ultrasonic washers are a system designed to generate and distribute ultrasonic energy. So the most important one is the stainless steel tank. It holds, let me just, yeah. The stainless steel tank holds the cleaning solution and the instruments, of course. And of course, that instrument will not be held directly in the tank. We have the basket that will hold the devices and instruments to be cleaned, which is a nest, a basket that will allow the sound waves to pass through the holes of the basket. So it's very important to consider the material of the basket so as not to reduce the cavitation energy that is reached by the instruments. The most important part of the ultrasonic washer are actually the transducers that normally in the conventional configuration of the oldest versions of ultrasonic washers are below the tank. Of course, with the advancement of different technologies, nowadays there are some manufacturers that also offer transducers on the walls of the tank, and especially those that offer a multi-layer or multi-level configuration. But generally, transducers are at the bottom of the tank, and they convert electrical energy into vibration of the water of the washing solution in the ultrasonic range. And this is why they are called ultrasonic washers, okay? So basically, most of the ultrasonic washers work within a specific range of frequency in the ultrasonic range so as to work and to deliver the right energy to the solution and create the cavitation that we need for cleaning our instruments. And last but not least, the cleaning solution, of course, that will harbor or will contain water and the chemical solution used for cleaning, the detergent. So how is the mechanics of the microscopic scrubbing that we have inside an ultrasonic washer? So ultrasonic sound waves created by the transducers create alternating cycles of high and low pressure inside the fluid, inside the washing solution. So this is what happens. First, we have the formation. Yeah? Cavitation is actually a translation of cavitation is the creation of vacuum bubbles, not air bubbles, but vacuum bubbles, void bubbles. So this happens because of this low and high pressure created inside the water that will create these cavitation bubbles that I will explain next. Okay, so how is the formation? The water expands. Okay, it separates. Well, the liquid molecules are pulled apart, creating these microscopic bubbles. After the inversion happens, the second step is the inversion. So the pressure starts to change, and this bubble starts to fall inwards. The third step is the jet extension. The high pressure makes the surrounding liquid pressure to make this jet that collapses the bubble, extending a blue liquid jet through the center. So it starts by creating the bubble, going inward, jetting inside, and then the implosion. So a microjet of liquid is blasted against the target surface, dislodging the soils. So this circle happens all the time, and is thanks to the ultrasonic waves created by the transducers that provoke this inside the solution, the washing solution, where we have water, where with everything that can be dissolved in the water, and including the detergent. Okay, when I talk about the water, I'm talking also about the air that is trapped inside the water. I'm talking also about the salts and the minerals dissolved in the water. So anything related to water quality will also influence in the this capacity that the water solution has to provoke this cavitation. Of course, we are talking about something very microscopic. This is something that is not able to be seen by the naked eye. We need a microscope, or we need something for corroborating that this is happening correctly, that will translate this creation of cavitation into something that we can read and interpret. Okay? Of course, as any other system or process that relies on all, that has to be effective, rely on different parameters in order to have the appropriate influence of energy, high and low energy, to provoke the creation of these cavitation bubbles. And the three main, besides the water quality that I have just mentioned, you can imagine that if we have to separate the water molecules to create these vacuum bubbles, anything dissolved in the water will influence this creation. So we need to make sure that everything else happening to the water solution is also under control. Right? The first parameter that I have introduced before, and is one of the most important ones, is the frequency, the working frequency. And this will be delivered by the transducers. Okay, so this is the main job of the transducers. And this is very important because inside an ultrasonic washer, we will definitely need every transducer to be working correctly. And when we have a big ultrasonic washer, we have dozens of transducers depending on the size, okay? And when we start a machine, we can hear the sound of the transducers, but it's very difficult to identify if one of them could be broken. So you can see that it's very important because depending on where my medical device will be located inside the ultrasonic washer, if the transducer at that location is not working correctly, the cavitation energy at that position will not be the precise that we need for the right cavitation energy to clean my instrument. So this is why it's very important to know how my ultrasonic washer is working. Generally, 40 kilohertz, between 35, 45 kilohertz is the normal working range of frequency of most of the healthcare dedicated ultrasonic washers. So that's the frequency of working of transducers work. Temperature is also very important because we need an equilibrium. If the temperature is too low, the vibration of the molecules is less, the movement as anything. Less temperature, less movement. High temperature, more vibration. So it's easy to separate the water molecules to form the vacuum bubbles. But too much high temperature, too high temperature can provoke, it can be detrimental, and we will see in the next slide. It's not too good for the cavitation to be as effective as we want. Okay, so we need an equilibrium of temperature. These implosions that I have explained in this previous slide, these implosions create very high temperature in the location of these implosions. So we need to be sure that the temperature does not rise above the necessary range, okay? Normally, between 50 and 60 degrees is a desired temperature. And last but not least, the chemistry. When we talk about the chemistry, again, the water quality is very important because everything dissolved in the water will affect the efficiency of cavitation, but also the detergent of our choice. The detergent need to be definitely low foam because the foam will be very detrimental to cavitation energy because it's a competition between the cavitation or vacuum bubbles with the air bubbles created by the detergent foam. Okay, so low foam, definitely. And we also have to make sure that we use appropriate detergents that the manufacturer states that are compatible with ultrasonic processes. Because the same energy delivered to clean instruments and to remove organic residues from the instruments could not be good for the efficiency of the detergent, even more if it's an enzymatic one. So we have to make sure that our enzymes in the detergent will stand for cleaning, will stand the cavitation delivered to the system to clean our instruments, and we will not inactivate these enzymes just because we are making an ultrasonic process, okay? So we have to make sure that we use also the right dose of detergent. More detergent is not always more effective or increasing the effectiveness of a washing process. We have to follow the manufacturer's instructions. Okay, so as you can see, ultrasonic washers have a delicate and easily disrupted balance. Okay, so just turning on the machine and hearing the sound of transducers working and seeing some waves on top of the solution does not mean that everything is working correctly and that the cavitation energy is necessary to clean my instruments. Okay, so we have to make sure that every day, every time we change the solution, we have a new system. Okay, so what was maybe working in the previous cycle could not work this time. Many things can happen. And even more if we change the solution to a new one. So the first thing that we have to do when we change the solution of our ultrasonic washer is the degasification, okay? Because we have to make sure that we remove from the water all the trapped air that is normally within the liquid water, okay? So we have to eliminate this because it's competition between the air and the vacuum bubbles that we want to create, okay? So we will eliminate this air through normal air bubbles, okay? So we will see when we degas the solution or the trapped air that will come out as air bubbles. This is the degasification. It is mandatory. Everyone should do this. When you put a new solution, you just have to run a degasification cycle, and then only after this, you put your load for washing. Okay? Of course, what will affect the efficiency is the load and the frequency of the transducers work, how the transducers are working. If I put heavy load or I put too much load inside, I will obviously be decreasing cavitation energy delivered to all the instruments. So overloading, of course, is a disadvantage. And the incorrect performance of the transducers, not at the right frequency, will also be detrimental for the cavitation energy. As I have mentioned, temperature too high will create vapor pressure, and this will destroy these cavitation bubbles. And of course, the basket design and material, okay? Not every material is compatible with an ultrasonic washer working with the right energy. Normally or generally, plastic baskets will absorb a lot of energy. So we have to make sure that if we are using plastic baskets, they are suggested or delivered by the same manufacturer of the ultrasonic washer. But it is very important to corroborate that even using this type of designs, we have a good development of cavitation energy. So you can see that many things can create dead zones because the cavitation energy and the ultrasonic energy delivered to the solution as high and low pressure waves run through the solution, and everything inside will interfere even more the load, okay? So we have to make sure that we can monitor and safeguard as many parameters as we can. So how much is enough? We are talking about energy. So how much is enough? So the optimal energy will be the one we are in the middle, of course, and it's the one that thoroughly cleans the complex geometries and blind holes without affecting the materials of the medical device that I'm trying to clean. So excessive energy is also a disadvantage because it can provoke permanent damage to our medical devices. Even more microsurgical, optical, and fiber optic instruments. Insufficient energy, of course, is something that we want to avoid because it leaves organic residues in our medical devices. Okay, so we need to find the optimal energy range to make sure or to guarantee that our instruments are being correctly cleaned. So consistent performance requires verified practices, not assumptions. Historically, CSSDs have relied on rudimentary tests. These methods only show if cavitation exists, not if the energy is at the correct, safe level for modern medical devices. The industry needs a standardized measurement. Okay? Of course, there is a global compliance matrix. Here, I'm just showing a couple of examples, but different regions have different recommendations, and some of them are mandatory. So just a few examples of the USA, which is normally globally adopted, the AME ST79, explicitly requires daily cavitation testing whenever the equipment is in use. The Joint Commission, which is also something that is applied worldwide when we have audit and we want to be certified by the Joint Commission, includes ultrasonic cleaners as mandatory inventory equipment. And so it requires testing. Australia is the only country worldwide that has a specific standard regarding ultrasonic washers and how to monitor them. And it mandates daily testing. And just a few examples in Europe, the ISO 15883, we say Europe, but it's actually an international standard. And the CRINCO, German recommendations, mandates validated automated cleaning procedures and periodic requalification. So these are just a few examples. We also have HTM. We also have APSIC guidelines. There are many localized recommendations so as to monitor, to be sure that our ultrasonic washers are performing correctly. Not all of them, of course, mandate to do this on a daily basis, but it is important to notice that standards talk about this and that the washer needs to be validated. So cavitation is completely invisible to the naked eye, as we have spoken. Passive cleaning efficacy is insufficient, so the actual presence of uniform ultrasonic cavitation energy should be validated. So the regulatory bottom line is that the absolute invisibility of cavitation failure is the exact scientific rationale of suggested to mandatory daily verification. So what is the solution that Terragen has developed in order to make this monitoring very simple, very straightforward? Is the ultrasonic cavitation performance test, for which we have two different options. We have the CDW and the CDWZ. We will mainly talk about the CDWZ just because both of them are very similar to each other. There is no performance difference between them, only what is contained inside the ampoule that I will explain now. But just between them, they have just different materials with different shapes. But it is exactly the same indicator with exactly the same performance. Okay, so the energy delivered, the cavitation energy delivered to one or the other will be seen exactly in the same way. Okay, so as you can see, what it is, this ultrasonic chemical indicator. It is a glass ampoule filled with a blue reactive solution, transparent, that contains also, in this case, ceramic cylinders in the Z version. In the CDWU version, it is glass beds or glass pearls, okay, round. These are cylinders of ceramic material. As you can see here, they can work within a range of temperatures, which are the cover most of the ultrasonic washers in the market, from those that work at room temperature to those that work at high temperatures due to specific use, okay? With ultrasonic washers working beyond 35 kilohertz, which are also basically all healthcare ultrasonic washers work at this range, and they can be used both for periodic functional tests as well as for routine tests. Okay? And our suggestion is to use them with this hook or holder that will make the uh, will attach, let's say, the indicator to a specific location, avoiding the movement of the indicator due to or as consequence of the vibration of the solution. Okay, so by using this hook, we just make sure that the indicator will not move very far away from the initial location. As you can see here, the indicator will change from the initial blue color to yellow, which means that we have delivered the right cavitation energy at the position where this indicator was located. Okay, and this is how it works. We have the initial blue color and the successfully processed or right cavitation energy delivered to that indicator. And in between, in the process, we will go through different shades of green. Okay, so the vibration and the energy delivered to the initial indicator at the location it's positioned will trigger, and with the right energy will trigger a reactive a chemical reaction, thanks to the vibration of the ceramic cylinders. And this chemical reaction will move from blue to yellow through the different shades of green. So if we don't arrive to the final yellow color, it means that the cavitation energy delivered to that indicator was not enough to guarantee the cleanliness of our instruments. Okay? So that's the concept. So as I have mentioned, we can use the indicator both for routine tests as well as for periodic functional tests. Of course, the number of indicators we use will be related to the size of the ultrasonic washer. The bigger the ultrasonic washer, the more indicators we will use, because we need to make sure that we will be corroborating the performance, at least the performance of every transducer during the periodic functional test. So we can use this indicator not only for making sure that on a daily basis we are delivering the right cavitation energy, but also for during the periodic functional test, we will map the tank and we will know the cold and hot spots of cavitation that my ultrasonic washer delivers in the full volume of the ultrasonic washer. Okay, so for routine tests, and what is a routine test in the case of an ultrasonic washer is every time we change the solution. Fingers crossed, this should be at least once per day, because the more cycles we do, the more organic residues we will have in that solution. So the ideal would be one cycle, change of solution. But if we don't do this, at least we should do this. And of course, it will depend on the level of organic residues or dirt in my instruments and how many cycles we perform, okay? But the idea will be to change the solution after every cycle. But when we change the solution, we put the water first, treated water ideally to preserve my instruments and the the the, the metallic part of my ultrasonic washer, and also the detergent at the right dose. We deliver or we wait for the temperature to be reached, and then we perform the degasification. As I have mentioned, this is mandatory. After degasification, we will put a number of indicators depending on the size of the machine, and only after we have corroborated that the ultrasonic washer is working correctly, we will load our instruments. Okay, so it's change of solution, degasification, testing, load. Okay, so I like to say, and I think this is something very easy to keep in mind, what is the CDWU or C doing to our ultrasonic washer? I like to say that they work as a Bowdick test. Okay, we make sure that our ultrasonic washer is delivering the right energy, the right cavitation energy to clean my instruments. In the same way, we use our Bowdick test to make sure that our steam sterilizer is eliminating the air and our steam is of the right quality in order to penetrate our items before we even put our load. Okay, so this is how the cavitation indicator works. So we will place 1 to 3 indicators depending on the tank volume. We will rotate positions. Here are just some examples, okay? But we have to make sure that the corners normally are the cold spots due to the configuration, as I have mentioned, the waves of energy run through the tank, okay? So the walls are generally, the corners, sorry, are generally cold spots of cavitation. So this is why it is important to test for the corners, but changing location of the indicator in the same way that we should do that with cleaning indicators inside thermal disinfector machines, changing location to corroborate that the full machine is working. This is the same concept. Okay, and to read the results after or within the first 30 minutes after the exposure, because the colors can revert. Okay, so the indicator should be used and tested or evaluated the result within the first 30 minutes after we finish the process, the cycle. And the periodic functional tests after acquisition, major repair, relocation, okay? And in this case, in the periodic functional test, we have to test all transducers. If we have 12 transducers, we have to locate one indicator on top of the every transducer. How we know this? Because we have the manual of the ultrasonic washer. The manual of the ultrasonic washer will display precisely where every transducer is located. Okay, this is mandatory information from the ultrasonic washer manufacturer. Okay, and of course, we have to document all the results in the qualification protocol or file. Okay, so this is how we use it. But which are the current challenges of the interpretation of these indicators, regardless of being the U or the Z? First of all, and the most obvious one, the limits of the human eye. The subjective interpretation of the visual reading makes a dependence on individual judgment, leading to variability and inconsistent evaluations of cavitation energy. And of course, as a consequence, we have the operational uncertainty. Without a standardized criteria, staff may hesitate when making decisions about process performance and corrective actions. And also the lack of traceability. Mm? Visual readouts do not generate documented records. So once the indicator is thrown away, the proof is gone. And you can see from the shape of the indicator, even though it will definitely revert at some point, even if it does not revert, keeping a record of this type of glass ampoule, it's very disadvantageous, of course, in every sense, okay? So this is why we will not keep a record of the glass ampoule itself, okay? So this critical flaw limits the trend analysis, the audit support, and long-term process monitoring required in modern health facilities. So what is the solution that we offer? The Bionova CaviTest device, which is the CDWZ and U automatic reader. Is the first system designed to standardize and digitalize the ultrasonic washer performance. And we will see how it works and what it does. As you can see in the picture, is a very simple device with 3 positions. So every position can be used for reading a cavitation indicator. We don't have an incubation time. It is instant. So this is why we talk about an auto reader and not an incubator, because we don't have to incubate. It's just an automatic reader. We have 3 positions. Every position will independently read an indicator. We will see how. For now, we will have 2 programs. One is only activated, and then we have different buttons that I will explain what they do. But just for you to have a quick overlook of the auto reader. Okay, so this auto reader will allow you to change the visual interpretation into quantifiable data, which is the aim of the machine and the objective of the webinar. So this machine delivers objective reading, quantifiable cavitation index, clear pass-fail results, documented traceability, and standardized decision criteria. No specific regulation exists, but best practices define excellence. So this is what we want when we offer our products, and this product is not the exception. So going back to the components, we will see after how it works and how it reads, okay? But the basic components, as I have mentioned, 3 reading positions and every activation button for independent reading and interpretation. The program selection button, the built-in thermal printer, a reprinting button. So if we want to reprint a ticket, we just press this button for reprinting the last 3 readings. The paper tracking button, just if we want to take a little paper out before tearing it out. Configuration button for changing language, mainly. And the lights, okay? We will have a red light with an X if we have a fail result or low cavitation energy. We will see how. And a green light with a tick when we have a cavitation index with that is showing a high level of cavitation energy. So how it works? Yeah, the CaviTest auto reader detects the final color of the CDWU and CDWZ through precise light transmittance measurement. So when we put the indicator in every position, the system will analyze the RGB or red, green, blue transmittance of that color. the system changed this RGB number into HSV parameters. HSV is hue, saturation, value, or brightness. Okay? So the RGB precise color interpreted by the machine is converted to HSV, and this, through a correlation, we have a number that we call cavitation index, okay? It is converted through an algorithm, RGB to HSV, and we will have a precise number called cavitation index that we will translate the color of the indicator to a precise number to decide if the cavitation energy is enough or not to guarantee the removal of organic residues from my instruments. Okay, so this cavitation index range is from 0 to 130. 0 is blue, non-exposed. 130 is the brightest yellow we can have after exposure to cavitation energy. In that range, from 0 to 100, we have, so from 0 to 100, we will have a failure. If there is a color deviation, this will be the colors displayed by the green shades, okay? The greenish colors between the blue and the yellow. From 0 to 99, actually. From 100 to 130 are yellowish colors that will display a good cavitation energy. Okay, so we translate these very difficult to interpret shades of light green or precise yellow. We translate it thanks to a precise calibrated device and an algorithm that will transform this color interpretation into a precise cavitation index. Okay, so with a single numerical value, we remove the ambiguity, we support clear decisions, and we enable trend evaluation. This is how the ticket will display the results. So you can see that we have clear pass and fail results. In the ticket, you will have, as any of our machines, the identity of the auto reader, in this case, a Bionova CaviTest, the batch and serial number of the device, the ticket number from 0, the first incubation, and is cumulative. The program being used, in this case, nowadays, we only have one activated program for both indicators. Of course, the position being used for the reading, the date and time of the readout, the result, if it's a pass or a fail, and of course, the cavitation index I have just mentioned. Okay, so you can see here 110 is a pass. It will correspond to an accepted yellow color, and 79 is obviously less than 100, which means it is a failure, and we will also have the fail result printed. Okay, so the pass is a cavitation acceptance criteria met. The ultrasonic washer may be used as normal. Fail, the cavitation acceptance criteria is not met. So you have to follow the established departmental procedures for the ultrasonic washer and decide what to do with the machine. As you can see here, it's just 1, 2, 3, really, okay? We just take out from the washer the ultrasonic cavitation test. We just clean it for the water not to wet the reading positions within 30 minutes to make sure that the color will not revert in the meantime. And we just position the indicator in inside one of the reading positions. We press the number, the bottom of that position, and automatically, immediately, with no incubation, we will have the reading of the result, as I have just shown you, like this, a printed ticket. Of course, as any other of our electronic devices, this device can also be connected to our softwares, and the record can be also digitalized for trend analysis. So visual interpretation is no longer enough. And Bionova CaviTest introduces a new best practice for CSSDs: objective data, consistent decision-making, and traceable results. Standardize your ultrasonic cavitation monitoring today. So we have the old standard, which is the visual approach, where we have subjective interpretation, undocumented results, and high operational risk, while we offer the new standard, which is the Bionova CaviTest option, with objective automated reading, fully traceable printed data, and consistent decision making. So the CSSD process maturity model is what we are trying to offer to the market, okay, where in this case, specifically for ultrasonic washers, we have the level 1, which is the soaking tank, high risk. We are running ultrasonic equipment with zero verification, relying on blind faith in the machine. We just hear it's working, but we don't know actually if all transducers are working and if the cavitation energy is necessary to clean our instruments. Then we can jump to level 2, the visual guess, which is compliance. We are compliant, but we are using chemical indicators, only relying on unrecorded, highly subjective human color perception. If we jump to level 3, which is the one we are offering nowadays with this webinar with the Bionova CaviTest, we have the quantified baseline. We are under control. We are utilizing the CaviTest for objective digital pass-fail logging and automated printed audit trails. And if we want to go to the top, to the level 4 with the predictive maintenance, gaining mastery in our monitoring, the tracking the cavitation index over time will help me to spot declining cavitation index values and replace transducers even before they fail. So if we continuously monitor on a daily routine basis how my machine is working, mapping our tank with indicators, changing positions, we make sure not only that today my machine is working, but that today my machine is not working, not reducing the energy or the effectiveness that it should deliver to my instruments. So just by measuring the cavitation index, we can monitor over time how my machine is working. And if we see a reduction in the cavitation index in specific positions, we can make preventive actions in order to avoid major maintenance because some transducer breaks down, okay? So CDWU and CDWZ make it visible. Bionova CaviTest makes it questionable. Validate your cavitation, control your process, protect your instruments, safeguard your patients. Thank you. And I will reduce the screen, but here you have, if you scan here, this QR code, you will go directly to the certificate of participation request. You just have to fill in a few questions. It's a very, very, very short and simple form. And just by filling that form, you will have the certificate of attendance sent to your email. And of course, if you want to go for search for more information, if you scan here, you will go directly to the homepage of the CaviTest within our website. Okay? I will just leave it for a couple of seconds so that those interested can scan the QR code. I will just reduce the screen so that I can see you, okay? But I will leave this screen so that you can keep.
Thank you, Anto, for the presentation. It's truly a great product. And I think that it's a groundbreaking product. It makes the whole difference between just estimating a result, like all color changing indicators. There is a subjectivity that with this product we solve that huge problem. We have a question by
Yeah.
Yeah, can you see it?
Yeah, it's from Nayana.
Yes.
Okay.
Correct.
Okay. How many indicators need to be placed for a 150-liter tank capacity? Okay, I will stop sharing, but after we finish, for those that are still interested in seeing the, in having the certificate, I will place the QR code again, okay? So this is not a straightforward answer. There is no straightforward answer because it does not depend only on the volume, but also on the transducers. So we should place a minimum of the number of transducers we have in that washer. Okay, so it's not only related to the size. Of course, the bigger the ultrasonic washer, the more transducers we will have, right? But this will depend on the manufacturing of this ultrasonic washer. So what we have to do to decide how many for qualification or periodic verification, we have to go to the manual of the ultrasonic washer and corroborate how many transducers we have and where are these transducers located. So for qualification, we should at least place one indicator in every transducer on top of every transducer location. This is for qualification or periodic functional tests, okay? For routine tests, we will already know we have our 3D or 2D, let's say 2D map of our ultrasonic washer. So we will not need to use one on top of every ultrasonic transducer, but we have to make sure that from cycle to cycle or from routine test to routine test, we will be changing locations so as to, in the end, map down all transducers. Okay, is it clear? So let's say, regardless of the volume, I have a machine, an ultrasonic washer with 15 transducers. When I perform the periodic functional test or the qualification of that machine, I will place 15 indicators on top of the signaled position of the transducers by looking at the manual of the machine. That's for periodic functional test. When I'm performing routine test, if it's a very big machine, for routine test, I would say at least 3 ampoules in distant locations, let's say middle and the 2 corners, and the next routine test changing these locations, okay? Always trying to place or to design the locations thanks to the mapping that I have of my of the manual of the machine. Okay, is it clear? Nayana, just let me know if you want to write down if it's the answer is clear, or if you have more questions about this.